This protocol allows us to easily explore the epigenetic features of neurodegenerative disease. The main advantage of this technique is that it exploits yeast as a model system. Compared to other cellular and animal models of neurodegenerative disease, yeast is expedient and cost-efficient.
This technique allows us to map out the epigenetic changes that arise in neurodegenerative disease, which may lead to novel markers for diagnosis as well as new targets for therapy. This method allows further investigation of the role of epigenetic marks in neurodegenerative disease and can be modified for the assessment of human fibroblasts and induced pluripotent stem cells. This particular method contains a number of details that need to be demonstrated visually, such as the proper loading of the Western blot apparatus.
Demonstrating the procedure will be Michel, Fallah, and Navin, Rana, both undergraduate researchers in the Torrente laboratory.
Begin by re-streaking yeast from frozen glycerol stalks onto histidine 2% glucose agar selective medium plates for a 2 to 3 day incubation at 30 degrees Celsius. At the end of the incubation, inoculate re-streaked yeast for each overexpression model and control in 5 milliliters of histidine selective liquid medium, supplemented with 2% raffinose at 30 degrees Celsius and 200 rotations per minute overnight. The next morning, grow 100 milliliters of liquid culture for each of the overexpression models and controls in selective medium supplemented with 2% galactose overnight. And measure the optical density at 600 nanometers, or OD600, to determine the volume of overnight culture needed to render overexpression cultures at a starting OD600 of 0.